Okay, today we're going to be talking about mitosis. Mitosis is a way for cells to divide, and in the process of that division, they produce genetically identical daughter cells. And so by genetically identical, we mean whatever amounts and numbers of chromosomes are in the mother or the parent cell are going to be in the daughter cells. So before that has to happen, the DNA needs to reproduce. So normally our cells in our body are diploid, and those diploid cells will go through a phase of interphase called S phase. S is short for synthesis. And then, that's the cat, Charles Perwin. And then this cell with 92 chromosomes in it will make 2 new nuclei, each with the 46 chromosomes that one of our normal cells needs. And then that cell will divide into 2 cells with 46 chromosomes each. So these daughter cells here are the same as the parent cell up here. This happens when your cells need to repair damaged cells or tissue. This happens when you need to grow. And this happens when you're doing some kind of development and you need to produce new kinds of cells, like in puberty or when you are developing in your mother's womb. So, the cell cycle itself can be divided up into almost all interphase. The first part of interphase is G1. That just means growth. That's when the cell grows a whole bunch, lives its life, does its specialized task, whatever that is. Then during the S phase, the DNA replicates. Of course, we know that it replicates semi-conservatively because we're all experts in Messelson and Stahl's experiment, right? Okay. Then we have G2 phase, and at this point, the cell will grow some more, get ready to divide. So all of these are interphase. That's where the cell spends most of its life. Once the cell enters mitosis, in a series of 4 steps, we'll abbreviate them for now, P, M, A, and T. In a series of 4 steps, it divides the nucleus into 2, which is what we did right here. This is the whole mitosis part. And then once it's all done with that, it does this last part called cytokinesis, where it actually splits in half. Cytokinesis. I'm writing with this big fat stylus right now. Sorry about the big mess. There's lots of checks that the cell goes through along the way, and if any of those little checkpoints aren't met, the cell won't divide. But let's start. We're going to draw a cell that has an N of 3. This means how many unique chromosomes it has. And by unique, we're talking about size and types of genes. So if we say we're going to have an N of 3, we're going to draw a long one, we're going to draw a medium one, and we're going to draw a short one. This cell is going to be a diploid cell, which means it has 2 of each chromosome. And it has 2 of each chromosome because one is a mom and one is a dad chromosome. That's where they came from. All right, so for example here, in our nucleus, here's our cell, and we've got our nucleus here, and I will draw a nice long chromosome, medium chromosome, and a short chromosome. But remember, this is a diploid cell because we had a mom and a dad. So here's our long dad chromosome, our medium dad chromosome, and our short dad chromosome. So our N is 3 because we have 3 unique chromosomes. long, medium, short. And we have a diploid cell here, which means we have 2 of each chromosome. So how does mitosis work? Well, we start here in G1 of interphase with a cell. And in this cell, we are making, once again, an N of 3. So we have our long chromosome, our medium chromosome, and our short chromosome. Then, since we have a diploid cell, here's another long one, here's another medium, here's another short one. Where they are, it doesn't matter. Some people get into the habit of making their cells have their DNA just kind of all sitting right next to each other like this. And I gotta say, that's a little bit of an immature sort of way to approach it, because your DNA is kind of spread out all through your nucleus. So I'm gonna recommend you don't do that. Okay, and then S phase happens, and in S phase, our DNA doubles, it replicates, because we need to get ready to divide, and we want to pass all of our chromosomes on to our next generation. So instead of having 1 long mom chromosome, we're going to have 2 long mom chromosomes, 2 medium mom chromosomes, and 2 short mom chromosomes. And we are going to do the same thing for the dad chromosomes. 2 long, 2 medium, 2 short. Once again, this is G2. Our cell passes all of its checkpoints. It's ready to divide. So it goes into mitosis. Now, the first stage of mitosis is the most complex one to draw. And in the first stage of mitosis, our nuclear membrane breaks down. These little cylindrical things that are at right angles called centrioles form and start to make some of these things called spindle fibers come out. These are going to be used to line up and move the chromosomes around. And then our chromosomes pair up. The 2 long ones that are exact copies of each other will pair up. The 2 short ones, the 2 medium ones that are exact copies of each other will pair up. This is prophase of mitosis. And we have a name for these different kinds of chromosomes. So let's draw our 2 long ones here, both mom and dad long ones. Now, the fact that these 2 are the same size and have the same kinds of genes makes these 2 chromosomes homologous chromosomes. That's when you have a maternal and a paternal pair of chromosomes that are the same length and have the same genes in the same places. But these other 2 here that are exact copies of each other aren't homologous chromosomes because they are not a maternal-paternal pair. These 2, since they're exact copies of each other, are what we call sister chromatids. But let's move on. So, let's switch colors here. I'm getting pretty good at this. All right. So, once our nucleus is totally broken apart, our centrioles are completely at opposite poles of the cell. Those spindle fibers will have moved all of our chromosomes, all of our pairs of sister chromatids, to the middle of the cell. Once again, sometimes you'll hear a globe analogy here. This is the equator of the cell. And they'll be all nice and lined up here. And our spindle fibers will be connecting to the centromere of each of these chromosomes. I really encourage you, after you take some notes and things like that, you watch some videos, see what questions come up. This is metaphase of mitosis. After metaphase of mitosis comes anaphase. We've lined all of our chromosomes up on the equator of the cell. Our centrioles are still on opposite poles of the cell. And our chromosomes, I forgot which order I put them in. Okay, our sister chromatids are now split apart and they're pulled to opposite ends of the cell. This ensures that each end of the cell gets one set of each chromosomes that it's supposed to have. Without doing this step right here, it might not guarantee that it gets one complete set of maternal and one complete set of paternal chromosomes. Finally, we move on to the last phase of mitosis. In this phase of mitosis, cytokinesis starts, which is not a phase of mitosis, but it happens at the same time. So our cell will start to stretch out here. Our nuclear membrane will reform. We can redraw our centrioles over there. I don't think we've gotten into that habit, but that's okay. And each cell now has a nucleus with a paternal long, a paternal medium, and a paternal short, a maternal long, a maternal medium, and a maternal short. Finally, we have cytokinesis. The cell will completely split in half. The nucleus will be fully formed. The DNA that's in there will start to unwind, but it's hard for us to really show unwound DNA without just drawing a whole mess of strings all over the place. So we're just going to draw it like that. And pow, we have two genetically identical daughter cells to the parent cells that are going to be used to repair or replace some tissue. The only other thing we need to write down is what cancer is. Cancer is when those little checkpoints get malfunctioning, and as a result, the cells reproduce like crazy. So you have a whole bunch of cells that are all sitting next to here, and normally you have what is called a tumor suppressor gene. Tumor suppressor gene. And when that gene is turned on, the cells reproduce just how they're supposed to. But when that gene gets permanently turned off, the cells go crazy and reproduce like mad, which is what a tumor is. It starts blocking off the function of your organs until eventually, and then eventually parts of the body stop working.